Hi, my name is Annika Pathak. I'm currently a junior in high school and I live in San Ramon, California. My presentation today is about the genetics of autism spectrum disorder. My mentor who guided me during this process is Carmen Maribel. I chose to write about this topic because my brother has autism spectrum disorder, and I've always grown up curious about how exactly this disorder affects him and how it affects his ability to think, and why he ended up getting it and not me. And my curiosity combined with my interest in molecular biology is what led me to write this paper. So first, we'll talk about the introduction, then the heritability of ASD, then the role of epigenetics in ASD, then the key indicators of ASD, and finally the discussion. So introduction. ASD affects about 1 in 59 children in the US. The prevalence of the disorder has increased 20-fold over the last 30 years. This is likely due to broader diagnostic criteria and recognition of ASD as a multiple causal spectrum disorder. ASD has a strong genetic component, with heritability playing a significant role. Certain DNA mutations disrupt brain development, leading to neurological and behavioral symptoms. Many individuals with ASD experience co-occurring conditions such as anxiety or epilepsy. Epigenetics is the study of how environmental factors influence gene expression. Maternal health and environmental exposures during pregnancy may alter fetal brain development. Factors such as chemical exposure and prenatal stress may increase ASD risk. This review will investigate the hereditary and epigenetic mechanisms underlying ASD, explore environmental factors affecting gene regulation in ASD, and analyze diagnostic indicators and variations across the autism spectrum. The heritability of ASD. Genetics plays a crucial role in ASD inheritance, with a 50 to 90% of risk attributed to genetic factors. If one child in a family has ASD, there's a significantly higher chance that a sibling will also be diagnosed. Siblings of children with ASD have a 20% chance of also being diagnosed, much higher than the general population, as aforementioned previously. ASD is linked to a heterogeneous collection of genes, meaning different individuals may have different genetic causes, leading to varied clinical presentations. Because of this genetic variability, a personalized approach to ASD diagnoses and treatment is essential, as each individual's genetic makeup affects both symptoms and treatment response. Twin studies provide strong evidence for ASD heritability. Identical twins are much more likely to both have ASD compared to non-identical twins. This supports the idea that genetics plays a major role in ASD's transmission across generations. While some cases of ASD arise spontaneously, the pattern of ASD in families is too consistent to be purely random. Some cases of ASD are linked to specific gene mutations, particularly on the X chromosome, which may explain why ASD is 4 times more common in males than females. Males have only one X chromosome, so they cannot compensate for an altered gene like females can with the second X chromosome. Other genetic disorders, like Duchenne muscular dystrophy, frequently co-occur with ASD, suggesting that some gene mutations affecting brain development may also contribute to ASD. Research has identified genetic variations on somatic chromosomes as well, particularly in the CDH10 and CDH9 genes, which are involved in cell adhesion and brain development. ASD may share genetic pathways with other disorders, like a reduction in acetylcholine receptors in the cerebellum, commonly found in individuals with ASD, has also been linked to epilepsy and schizophrenia. While the X chromosome plays a role, ASD is also linked to abnormalities in the somatic chromosomes, meaning it cannot be traced to a single chromosome. Duplications on chromosome 15 have been associated with ASD-like traits, including language delay, intellectual disability, and epilepsy. Up to 3% of individuals with ASD in studied populations have this duplication, but not all do, indicating it is one of many possible genetic factors. Chromosome 16 abnormalities are the most common specific genetic mutation linked to ASD, yet they account for only 1% of cases. More than 75% of ASD cases lack a clear single genetic cause, despite multiple smaller genetic linkages being identified. The fact that no single gene or chromosome explains ASD reinforces the idea that ASD is a complex, multifactorial disorder with contributions from both genetic and environmental factors. 3. The role of epigenetics in ASD. Epigenetics studies how environmental factors influence gene expression, effectively turning genes on and off. 40 to 50% of ASD variants may be linked to environmental factors such as drug exposure, toxins, and nutrition. Environmental factors can cause copy number variations, or CNVs, which affect gene expression and contribute to ASD development. Individuals with ASD have 3 to 5 times more CNVs than those without the condition. The three main epigenetic mechanisms that impact the risk of ASD are DNA methylation, which silences genes and reduces protein production, histone modifications, which alter how genes are packed and expressed, and microRNAs, which regulate gene expression at the RNA level. Studies on lymphoblastoid cells and whole blood DNA from monozygotic twins with differing ASD diagnoses have identified multiple differently methylated regions, or DMRs, between the twins and control samples. 400 DMRs were found in placentas of children with ASD compared to those of typically developing children, mostly in promoter regions linked to brain development. Methylation levels of two specific DMRs, the CYP2E1 and IRS2 genes, were linked to both the child's genetic makeup and the mother's prenatal vitamin use, highlighting how external factors interact with gene regulation. A possible ASD biomarker was identified. A CpG methylation site on the PPP2R2C gene was more methylated in ASD patients. This gene was found to be downregulated in people with ASD, suggesting that external environmental factors have influenced gene expression. Advanced parental age is a well-established environmental risk factor for ASD. A meta-analysis of 27 studies found that for every 10-year increase in parental age, the risk of ASD increased by 20%. Age-related changes in DNA methylation in sperm may contribute to ASD risk as well. Elevated prenatal testosterone levels are often observed in individuals with ASD, suggesting a link between sex steroid exposure and ASD development. Maternal smoking, substance use, and certain medications increase ASD risk. Inadequate prenatal nutrition can disrupt gene expression in the fetus, potentially contributing to ASD risk as well. The mother's health and habits influence gene regulation in the developing fetus rather than directly altering the child's genetic code. Pollutant exposure can alter the expression of genes involved in brain development, potentially disrupting normal neurological function. Notable environmental toxins linked to ASD include PCBs, lead, bisphenol A, mercury, and pesticides. PCB exposure alters DNA methylation patterns, impacting sexual development and gene expression in the brain. Endocrine hormone disruption has been proposed as a factor in ASD, potentially explaining why ASD is more prevalent in males than females. Epigenetics highlights how environmental influences interact with genetic predisposition, offering new insights into ASD development. Recognizing epigenetic mechanisms may inform preventative strategies and early interventions during critical developmental stages. The key indicators of ASD. Early identification is crucial to ensure that children receive appropriate interventions and support. Despite the genetic and epigenetic influences on ASD, behavior remains the most observable indicator. Parents are often the first to notice early signs, such as difficulties with social interactions and repetitive behavior. In 2013, the DSM-5 merged all ASD subtypes into a single category, emphasizing the need to recognize other developmental and behavioral issues that may co-occur with ASD. The DSM-5 criteria categorizes ASD symptoms into two core domains: social communication and interaction difficulties and repetitive behaviors. These criteria provide structured guidelines for identifying ASD, ensuring that specific behavioral patterns are recognized. Although ASD symptoms originate in the brain, they are primarily observed as behavioral differences. Symptoms can vary based on age, language ability, and cognitive development. Common behavioral indicators include difficulty understanding others' intentions, limited eye contact, and unusual use of gestures, and these behaviors contribute to challenges in social communication, pretend play, and interactions with peers. The consistency of these behaviors among individuals with ASD aids in early recognition and diagnosis. Co-occurring medical conditions can serve as additional indicators of ASD, reinforcing the need for comprehensive evaluation. Screening tools, such as the Modified Checklist for Autism in Toddlers or the MCHAT, help in identifying early signs of ASD, allowing for timely intervention. Monitoring developmental milestones is critical, as even subtle deviations in communication and social interaction may signal ASD. Family members play a key role in the diagnostic process because their observations provide valuable insight into the child's behaviors and developmental patterns. Sensory sensitivities are common in individuals with ASD, like heightened reactions to loud noises or certain textures. These sensory differences often coexist with social and behavioral symptoms, making them an essential part of the diagnostic evaluation. Recognizing sensory sensitivities strengthens the accuracy of ASD diagnosis. Discussion. Heritability is a central factor in ASD, highlighting the genetic basis of the disorder. Studies consistently show that ASD runs in families, with higher reoccurrence rates among siblings and identical twins. Environmental influences such as prenatal exposures and parental age contribute to ASD risk, and these factors can alter gene expression, reinforcing that ASD is not solely an inherited condition. The interaction between genetic predisposition and environmental factors is critical in ASD development, as environmental elements can modify how genes function. Epigenetic mechanisms significantly impact gene function and influence genes crucial for brain and nervous system development, potentially explaining the variability in ASD symptoms. DNA changes such as methylation play a pivotal role in both the onset and manifestation of ASD, and recognizing these genetic and environmental influences enhances our ability to better assess ASD severity and spectrum placement. These findings provide valuable insights into ASD's complexity, reinforcing the need for a multifaceted approach to diagnosis and intervention. Thank you for listening.